All right, in 17.2, we're going to talk about using immunology and immune molecules to do basically diagnostic tests to see if people have diseases and to see how much of a disease they might have in terms of how many microorganisms are in there. So we're going to talk a lot about this concept of immunoprecipitation. We can use that to measure immunoglobulin levels, so how much antibody is someone making. We can use immune molecules actually to do blood typing and to detect pathogens. And we're going to talk a lot about a specific type of immune assay called an ELISA. And then we'll talk a little bit about monoclonal antibodies and finally about immunofluorescent microscopy. Okay, so everything we're going to be talking about in this section has to do with the branch of immunology called serology, which looks at the contents of serum and is focused kind of a lot on antibodies and antigens. We're going to mention these two terms. We'll come back to them later in the course when we actually talk about a large number of diagnostics. But we have this term specificity. This measures how selective an antibody is towards an antigen. How well does it find a specific antigen? And then sensitivity is looking at how much or how many antigens or antibodies are present in a test in order to turn it positive. So we're going to talk about several different antibody type tests, some that look for antibodies and some that use antibodies to see things. But you're probably familiar with them if you've ever seen a home pregnancy test or rapid COVID test strip. These are all working off of principles using antibodies. And this one is very complicated. This is looking at different levels of IgG versus IgM. So you can tell, is someone making IgM only or IgG only or both IgG and IgM? This would be a tool to do that. And there's, of course, a control line that tells us that the test is still valid. We'll come back to these. We're going to introduce the principles of them in this chapter, but in chapter 25, we'll come back to these types of tests in a little bit simpler example. All of this is using immune molecules, particularly antibodies. And the ratio of antibody to antigen can lead to some interesting things happening. If there is more antigen than there are antibodies, the antibodies will be fully filled and there'll just be excess antigen floating around. If we have less antigen than antibody, not all of the sites get filled up. But if you have a precise ratio where the amount of antigen is equal to the amount of binding sites of antibody, the antibodies can form a complex here. And this complex can actually become so big that will precipitate or fall out of the solution. We call this immunoprecipitation. This can help us in several ways. We can use immunoprecipitation in this assay called a radial immunodiffusion assay to tell how much antigen is in there. So we could look at this, and it's not critical that we know this, but this is a gel with antibody in it, and we put antigen in that we're testing, and the more antigen that's in there, the larger the circle becomes until we reach the point where everything has immunoprecipitated. You can see this one has less antigen and it doesn't spread as far out. In this case, we're asking how many IgA molecules were in a sample, and we're actually using antibodies that measure antibodies, so it's a little blowing your mind there, I know. We'll look at some simpler examples here. Agglutination is a bit easier to understand. This is kind of like precipitation, but in this case, we're dealing with a liquid generally, and we're measuring insoluble antigens on like whole cells or something like that. So we could be looking at blood when we're talking about this, or we could not necessarily be looking at cells, but viral particles. We could be looking at whether someone has COVID-19 antibodies in their blood. So someone who has not been infected with COVID-19 will not have any antibodies in their serum, and these will not attach to particles that have COVID-19 antigen on them. But if someone's been exposed and has formed an immune response, they will make antibodies against COVID-19, and these antibodies will cause these particles to agglutinate, or basically clump up and form a clump. Okay, we can actually see this in a test, but let's look at a very easy, simple test to understand here. So this is for identifying microbial pathogens, but we can also use this to identify people's blood type. Blood typing, A, B, and O, has to do with what proteins are on the surface of your blood. We call these blood antigen types. There's protein A, and there's protein B. You could have one of those. You could be A, you could be B, or you could be AB, or you could have none of them, and we call that O. Now, interestingly, people who make type A blood antigens, so they have this type A on the surface of their blood, they actually make antibodies that react and bind to type B blood. So, if you are type A blood, you cannot receive type B blood because your immune system will attack it and basically cause an immune response to it. So you could get blood from someone who has A type blood or O type blood because they have no antigens. You can't get B or AB blood. The reverse is true for type B. They make anti-A antibodies. If you are AB, that means you have both antigens. You don't make any antibodies against these. And you can take type A, type B, type AB, or type O, so you are the universal recipient. If you are type O, unfortunately, you make anti-A and anti-B, so the only type of blood you can receive is O, but you can donate to anyone because you have no antigens on your blood. Now, we can test for this quite rapidly using a special IgM molecule that will react to either A or B. So we take a drop of blood and we put it on a glass slide, and we apply anti-A molecule to it and anti-B molecule to the other drop. You can see the anti-A is reacting to this blood drop. So this would be type A blood because it is agglutinating. The antibody molecule is binding to the A on the surface, and it did not do that in the anti-B test. So this would not be type B blood, and it's definitely not AB because there's no B reaction in here. So we can use it for rapid blood typing as well as identifying antibodies against microbes. There's a related technique that we call the enzyme-linked immunosorbent assay, or ELISA as it's commonly called. This relies on special antibodies that have an enzyme bound to them that basically, when it gets activated, it kicks off some sort of product, we call a substrate here, that's going to give us a color. And we can use how much of that color we see to quantitatively measure how much of this antibody is bound. In this example, we're looking at the simplest form. Well, I say the simplest. It's not actually the easiest to do, but is the least complicated, let's say. So this is called a direct ELISA. We have a glass plate here, and we take a patient's serum sample, and we have to bind antigens in that to this glass plate. There are various ways this can happen. But then we flush over an antibody that is looking for that antigen. If the antigen was present in the person's serum, the antibody will bind and it will give off that color. The more color, the more antigen they had. So here's an example. How many SARS-CoV-2 viral antigens were in someone's blood sample or swab? I guess a nasal swab would be a place we would find those. We would take that nasal swab, put it in a little bit of liquid, wash it over this plate, attach it to the plate, and then wash the antibodies over. The more COVID-19 SARS-CoV-2 viruses were in there, the more antibody would bind. Okay, that seems simple, but that getting it to attach to the plate part is actually kind of difficult. A different version of this is actually looking at not the antigen, but the antibodies. So this is an indirect ELISA. So here we have a plate and we put a known antigen on it. Let's say it's the SARS-CoV-2 viral spike protein. We could take patient's serum, so we could take your blood and wash it over the plate. If you've ever had COVID-19 or have been vaccinated against it, you will make antibodies against that, and they will bind to the antigen on the plate. Now, your antibodies don't have that special color attached to them, so we'd use a second antibody, we call a secondary antibody, that has the enzyme with the color attached to it. This is going to detect human antibodies and will bind to it. So the more color we get, the more antibody you had against SARS-CoV-2. So this would be something to see how well your immune system is functioning in this question. The most common ELISA, and the one that you're probably actually familiar with if you've ever taken the rapid test, is called the sandwich ELISA. Not because we're looking at sandwiches, but because we make an enzyme sandwich here, basically. On the plate, we put an antibody that captures a specific antigen. This could be the COVID-19 spike protein. We then use a secondary antibody to wash over that will bind to this and cause it all to stick together. This has a secondary antibody that gets conjugated to it, and with the color on it. And this whole sandwich will then make a color at this point. So the rapid test has this plate with all these anti-COVID-19 antigen antibodies on it, and then we wash it over, and in that strip are these special molecules with these enzymes that make the colored lines on the COVID test there. The control line is looking to make sure that these things made it all the way down to the bottom of it. We'll talk about that again later in the course. So the first and the last one are kind of doing the same thing, but it's just in this one we have to attach the antigen to the plate. In this one we don't, which makes it a lot simpler. That's why we can do like the rapid test strips. The middle one, the indirect here, is looking at how much antibody is present against an antigen. ELISAs often get used to measure what we call titer levels, which are the amount of antibody or antigen that is in a solution. So we can look at this, we could say, how many viral titers does this person have? This titer concept comes from a series of dilutions. Let's take an example where you have a patient with HIV. It's really critical to know how much virus is in them because that determines how likely they are to develop AIDS. So we take a patient sample, and we have 4 different patients here. And then we do dilutions of their serum, and then we do a special ELISA that is looking for the viral antigen, in this case, the HIV virus. The more we can dilute this and still get a color, that means that we started with a higher level of antigen. So this person in lane 4 has the highest level of virus in them because we can dilute it all the way down to 1 to 128 and still get color. This person in lane 1, when we dilute it past 1 to 16, we no longer see the color, so they had a lower viral level. This titer measure is quite common. ELISAs are very, very common tests for measuring how much antigen is in there, and by extension, how much of the microbe is there. So we talked about the sandwich one. Here's an example, a real life example. Say we want to figure out, does someone have hepatitis? We could use a sandwich method to detect that. So we would use an antibody that binds to hepatitis antigen. So we flow their serum over. If they have hepatitis, the antigen binds. And then we flow the secondary antibody over. That sandwiches it and secures that. Then we do one more antibody that detects this specific antibody, and it will make a color, and we can detect that. This is complicated, and to do this, we need special antibodies. We call them monoclonal antibodies. Monoclonal antibodies, you've probably heard of from COVID. We'll talk about that in a sec. But a monoclonal antibody is an epitope-specific antibody. So remember, it's specific to a very specific part of an antigen. And it's produced a single antibody producing B cell clone. So we'll see what this is. Polyclonal antibodies are anti-sera that contain many different isotypes. So IgG, IgA, IgM, all these different types. And they might not be as specific. They can act against different epitopes of an antigen. So they're a little less specialized. Monoclonal are like super targeted. To make monoclonal antibodies, unfortunately, we have to use animals, and the animals have to be sacrificed. But luckily, we can increase the lifespan of our production of this. So we do it once, and it lasts for quite a while. So we start with something like a mouse or a rat, or a rabbit is very common. Rabbits are very large, so they work quite well for making antibodies. We inject the antigen that we're interested in making antibodies against into the organism. That's going to stimulate the adaptive immune system, causing B cells to be formed. We then take those B cells out of the spleen where they will travel, and we fuse them with a special tumor cell. Remember, tumor cells will continue to divide indefinitely, whereas B cells will only do a certain number of divisions. So we fuse them together and form what we call a hybridoma. These are going to divide, and they will be little factories, and we can grow them in a dish, and they will produce our monoclonal antibodies against our antigen. And we can keep this cell line going for quite a long time. Not forever, but for much longer, so that we don't have to sacrifice another animal to do this. So we make these monoclonal antibodies using these cell cultures. And during the pandemic, we wanted to make monoclonal antibodies that would target the COVID virus. Because we didn't have any antiviral drugs that worked against SARS-CoV-2, we hoped that we could use antibodies to neutralize the virus. The idea is that we take patients that are sick with COVID-19, they have the virus in them, and we inject them with these antibodies. It will coat the virus and prevent it from replicating inside of their body. Unfortunately, it didn't work that well. The current recommendation, and this really hasn't been updated much because we kind of stopped doing it once we got some antiviral drugs, was you use these for high-risk patients with mild COVID. So that would be like the extreme elderly. So we saved these very precious antibodies for those individuals. This only works after someone is infected, so it's not like vaccination where it can protect beforehand. It's very expensive, and it really didn't work very well. Once someone was hospitalized with COVID and had severe COVID, the CDC and researchers found basically there was no benefit to using this. So they just stopped using it for hospitalized patients because it just didn't work very well. So in theory, it can work, but in practice, it didn't work very well. Finally, we can use antibodies to detect specific microbes. There are many tests that we call immunofluorescence tests, where we can use immune molecules, so antibodies, against a specific pathogen. So here we have Treponema pallidum, which causes syphilis, and we can attach what we call fluorophores to these to make them glow. So we could do a direct immunofluorescence test where we have antibodies against Treponema pallidum, and we could put these fluorescently labeled ones here. This requires a lot of work because the antibody has to be made against Treponema pallidum and it has to have the fluorophore added. So there's also what we call an indirect test where we have an anti-Treponema antibody. So this would come from the patient, and then we apply a immune molecule that targets any human immune antibodies. So the glowing one is universal to all human antibodies, so it will bind to it, and So many of these antibodies get bound that we can actually see the organism here, and we can see that it is the spiral-shaped Treponema pallidum here. So this is a great rapid test. It takes just a few minutes to confirm that this microscope slide has Treponema pallidum on it. Okay, a lot about antibodies in there, but they're so versatile, right? We can target them against super specific things, so they're super useful for all these different tests. Like I mentioned, we will come back and talk about those rapid tests in chapter 25. So if you're interested in that, look forward to that.